So what is the function of meiosis in multicellular eukaryotes? What are we doing? Why do we need to undergo meiosis in humans and other eukaryotes? Produce sperm and eggs, which has what function?
Reproduction.
Sexual reproduction. So meiosis is critical for sexual reproduction in multicellular eukaryotes. We can define sexual reproduction as a process that requires a fertilization event in which two gametes, a sperm and an egg, unite to form a zygote. When you get a sperm and an egg that fuse and fuse genetic material, it's called a zygote at that point. So here we have a mature egg, and here's the nucleus of this egg. Here's the shell, the zona pellucida, and then we have all these different sperm that are trying to penetrate into the cytoplasm of the egg. Well, here one sperm is able to penetrate, and it contributes its genetic material to this cell. So when we talk about organisms being diploid, half of our chromosomes come from our mother, half of them come from our father. This is the event where that's occurring. Half of your chromosomes are in the nucleus of the egg, and the other half are coming from the nucleus of the cytoplasm. This is the event where you get all your sets of chromosomes, all 46 chromosomes. So then they mix together. These are all different chromosomes here, the chromosomes from the nucleus and the egg, and what you get is a single cell called a zygote. A zygote, and this is a diploid cell. So here's that fertilization event that we were just talking about, and here's half a uterus. This is half of a reproductive organ in a female. So we have the uterus, the oviduct, and then we have the ovary. And what's going to happen is you're going to have a mature egg which gets ovulated out of the ovary, goes into the fallopian tube, and then as it ovulates, if sperm is able to make it up the uterus, up the fallopian tube, then the fertilization event usually occurs in the oviduct there. So that's where the zygote is formed. From that point, so here's our zygote. From that point, it undergoes cell division. 2 cells, 4 cells, etc., etc. What's the process that is occurring in which we have cell division after a zygote is formed?
Mitosis.
That's mitosis. Okay? So this is what I wanted to do here is bring meiosis and mitosis together. Meiosis is the formation of sperm and eggs, and then they fuse and fuse their genetic material, and then the organism is able to develop into a fetus and then into an adult organism with trillions of cells because of mitosis. All right. Michelle Simpson. How many chromosomes are there in every somatic cell in the human body during G1? How many chromosomes are in each one of our cells? 46 chromosomes. And we are considered diploid organisms. So how many chromosomes, if we're considered diploid organisms, how many chromosomes are there in a homologous set? 2. Right, diploid refers to 2. Where do each of these homologous chromosomes come from? One from your mother, one from your father. So here's a homologous set. It's diploid, so we have two chromosomes in that homologous set. One's going to come from your mother. One's going to come from your father. And we have 23 sets of homologous chromosomes. All right, so here's the human life cycle here. And if a zygote is diploid, it gets half its nuclear material from the egg, half the nuclear material from the sperm. And the zygote is formed by the fusion of sperm and eggs. Then how many chromosomes do each one of these sperm and eggs have to have? 23. What's the ploidy condition of those gametes? Haploid. So again, just to define ploidy number, we're going to define ploidy number as the number of chromosomes in a homologous set in a sexually reproducing organism, and in particular during the G1 phase prior to DNA replication. Okay, let's just make sure we have a handle on this. This can either be a somatic cell, or it can be a germ cell, which is a precursor to a sperm or an egg. So Whitney, this cell here is in G1. What's happening during G1 of the cell cycle? Normal day-to-day function. If it's a skeletal muscle, then it's contracting. If it's a nerve cell, then it's sending electrical signals throughout the body. This is where the cell does what it was meant to do. Brad, how many total chromosomes are there in this cell?
2.
There's 2. Michelle, how many homologous sets are there in this cell?
1.
There's 1. And how can you tell that there's 1 homologous set? What is it about those chromosomes that makes you think they're a homologous set?
They're identical.
They're identical with regard to length, centromere position. If we were to stain them and put them on a karyotype, they would stain the same. And they also have, what's the big one here? What's the other thing homologous chromosomes have in common? One's from the mom, one's from the dad. What about the genetic material? Timothy?
They are like opposite of each other. Like one's for blue eyes, the other is for the not blue eyes.
Alternate versions.
On the right track?
Alternative versions of the same gene?
Alternative versions of the same gene. So there's 1 homologous set there. Let's see, Lindsay. How many chromosomes are there per homologous set?
2.
There's 2. And Svetlana, what's the ploidy number of this cell? It's diploid. Why is it diploid? Brian, help her out.
There's 2 of each.
There's 2 chromosomes in a homologous set. All right, so this is during G1. After G1, we go through the S phase of the cell cycle. What happens during the S phase of the cell cycle? DNA replication. So each of these chromosomes duplicate themselves. What are these duplicated chromosomes called? Sister chromatids. So now let's do this same exercise, but instead of during the G1 phase, we're going to do it during the S phase.